Okay, so should we get started with your questions or the questions that came in through the Q&A, uh, the email and the personal messages? Okay, let me back up, give an introduction to what we are doing, why we are doing this, how we are doing this. Um, so basically, Ultragenyx was working on CDKL5 gene therapy. And if you even check their website, there are statements and
Yes.
notes. Uh, Go ahead, Kathleen. Okay. Okay, so if you see their website, there were statements, and there's a whole blog post that we wrote for Ultragenyx, and we were very excited about the gene therapy program that they were working on. Um, in October of 2022, at the CDKL5 forum in Boston, they announced the IND and beginning of clinical trials in summer of 2023. I met with Dr. Emil Kakis in summer of 2023. There was no announcement that they were starting this trial. So I asked him what was going on, and he mentioned to me that there were a few challenges that they are working on. So I was like, okay, yeah, that's fine. That's fair. Usually there is sort of a delay. Um, and then the same thing happened in summer of 2024. I asked the same question again, and this is when they told us that they've deprioritized the CDKL5 project for the other projects. And Basically, in fall of 2025, they were going to start the other gene therapy trial, but the FDA asked them for more documentation or got delayed. So what that means for CDKL5 is that our project has further gone pushed in the pipeline. Similar to that, like we've previously had Amicus uh,
Yes.
Therapeutics that
Only
was
five
working on
people
this.
here.
Yeah, I think,
Kindly go on mute, not speaking.
yeah. Like, if there are any questions, you can, virtually raise your hand in the Zoom, and then I can get to those questions. So it's since 2023, we are in end of 2025. We know Ultragenyx has paused the project. They haven't started it, or they haven't, given any more updates since then. And they've told us that they are prioritizing other projects, and once those are cleared up, they're going to restart this. Similar to that, when Ultragenyx announced the clinical trial back in 2022, the Lulu Foundation and their company that they had formed, Ilaj Bio, they also stopped or paused the work. And I had mentioned this to me. The reason was that like you cannot have two programs very similar to each other. Uh, um, that FDA may or may not approve both the projects. So from that perspective, Ilaj Bio, the company that Lulu Foundation has formed, they paused their project and they restarted the work now, and they are projecting clinical trials in 2027. So I had previously I was working on drug repurposing, and Ultragenyx was working on the gene therapy. So there wasn't any reason to start a new project or a third project for CDKL5 gene replacement therapy. So the drug repurposing, we've had success with that. We got the orphan drug designation. We are working on getting that to clinical trials with John Hopkins. So, like the work on that end is pretty much completed. It's just now a matter of John Hopkins starting the clinical trials and taking it from there, further from there. And in April, when I talked to Dr. Kakis, and in April of 2024, when I talked to Dr. Kakis, and I was like, why is this not moving forward? What is going on? So we started with a we started by understanding what the statuses of different gene therapy projects were at that point, at that time, and they were not necessarily moving forward. So we started the gene therapy program after understanding this, the landscape that this is not, the other projects are not going forward. And with the history that has happened through Amicus, it is it is known, or it is like kind of understood, that if it is not parents initiated, there is not necessary there's going to be a success with it, because for these pharmaceutical and biotech companies, they have a board of directors whose priorities are not the same, or they might not be similar to our priorities. So as a group of, small group of parents, we started this gene therapy project, and we tested it for efficacy in CDKL5 mice at two different ages. In older mice, in 3 months old mice, that is equivalent of a human adult, and a mice that was treated at day one of age, which is equivalent to a toddler, and we are seeing efficacious results in both of them. We had the pre-IND meeting with the US FDA. We proposed what our next steps are, what are we going to do, how are we going to do about it. We proposed the clinical protocol synopsis in there. The FDA provided us feedbacks that this is what you want to do, this is what is, is an optional step. It's like, we asked them that can we use pigs instead of non-human primates, or pigs instead of monkeys, and they clarified yes, you could use pigs as an alternate. They We presented to them the option of using a CDKL5 pig model, which is very close to the humans, in the sense that they show spontaneous seizures like our children do in very early age, as well as they have these issues or these survival challenges that is seen with other complications that happen with CDKL5. So the CDKL5 mice do not show the spontaneous seizures like the humans. So from that perspective, the mice is not a complete model. The non-human primates, the monkeys, we don't have CDKL5 model in that. So from all these different reasons, that's why this pig is the best model, which because we it shows CDKL5 deficiency symptoms like the patients with CDKL5. So that was that. So right now, as we are talking, we are doing the biodistribution and toxicology study, and like we are working on the next steps. Medica Biotechnology, this company that is based in Texas, is doing the manufacturing for us. Similarly, Charles River Laboratory is where we are doing the toxicology tissue analysis. University of Missouri, the National Swine Testing Center, is where these pigs will be treated, or where they will be injected with this AAV9 vector. This AAV9 vector is very similar to what Ultragenyx was doing, very similar to what the Lulu Foundation was doing previously. They've changed the vector slightly because Dr. Jim Wilson has a slightly modified version of the vector, but nonetheless, this AAV9 vector that we are using has extensively been used in neurological pediatric genetic conditions like CDKL5. So it is not novel from that perspective. It is replicating what others have done previously, and just encapsulating, just putting this CDKL5 gene in this AAV9 and delivering it to the brain. Okay, let's see. Is the, one of the questions that came up And I don't know, like for some reason, it seems like those questions got deleted or I can't see them anymore. But yes, one of the questions came up that who's this team, who's working on this, and so on and so forth. So my role as a parent is just to support in the financial structure as well as making sure that these different teams, like it's more project management, that these teams are talking with one another, but the actual work is being done at CROs, contract research organizations, as well as universities that specialize and do this work. Dr. Jacint Gingras, and her profile is on the website, and you can do a quick Google search and see her the details of what like the success stories of the previous genetic therapy projects that she's done. She is the scientific advisor to this project, so everything and how we move on to the next step and is vetted by her to make sure that we are not missing anything in terms of safety or efficacy. UCSF is where this clinical trial will take place. Dr. Nalin Gupta, he is a neurosurgeon who worked on AADC gene therapy. So, and he's the PI for this project. We are working with the centers of excellence and trying to figure out how to go about this, because there is a lot of back and forth with these organizations like Boston Children's Hospital, and from their aspect that they need to they need for these things to be vetted through their attorneys that they have. So we are in that process, and we are working towards making sure that all the pieces are in place. And if there are, we can have it peer reviewed, as well as this data is being worked on, being published. So those are the steps that we are taking. In terms of the financials, so we are a registered nonprofit 501(c)(3), meaning that we are obligated to follow all the IRS rules and regulations for nonprofit. The like all the expenses for these things are go towards the gene therapy project specifically. There isn't any overhead or salaries or anything that we are doing from the parents' perspective that they are involved in this. They are all volunteers. They are volunteering their time to take it to the next steps, and no, like none of the funds that being raised for this are going to the drug repurposing or any other projects that we've done in the past. It's specifically to gene therapy and only gene therapy. The major costs are the manufacturing of this injection that will be delivered, as well as the clinical costs associated with the patients when they are being delivered, and the monitoring after that. And the protocol return right now is such that these patients will be monitored for 3 years after the treatment. So I think the questions that came in via email and on the Facebook community, I think I've answered them all, but obviously there may be something else also that you want to ask. So let's take this opportunity to ask any questions, and we can work on that.
I have a question.
Yes.
Okay, just ask. Thank you so much for all you're doing. And is anybody duplicating this research now? Like UPenn, Jim Wilson, I've read a lot about him. Is Lulu working on the same thing? And where the heck is IFCR in this process?
Mhm. So that's a good question. So as I mentioned previously, we picked up where Ultragenics had paused, or so our project is very, or even other than the promoter, even the vector is same as Ultragenics. Lulu Foundation that had formed Elaj Bio, and they paused it when Ultragenyx had announced this project. They restarted the project as of fall of 2025. However, the vector that they are using is slightly different than the one that we are using and what Ultragenyx was using. So they are doing something which is slightly different, and they've restarted the work recently. And Lulu Foundation is projecting that it will go to the clinic in 2027. That's what they mentioned at the forum. It's slightly different. It's a different vector, so it's not that anybody is replicating this work or like Ultragenyx paused it. We are working on it. Lulu Foundations is a different vector. That's what the status is currently.
And why? So these people started with the vector that you're currently using and modified it. Why is it? I mean, there must be some reason they modified this vector to change it a little bit, right?
Of course, yes. So Dr. Jim Wilson is leading this effort, and he and his lab is the one that modified this vector. I believe it's the HU version, like it's slightly different version of this vector, and that's why they are proceeding with this, or it's Dr. Jim Wilson's lab and his uh, that's something that he's worked on previously.
Right. So I'm just confused. Why we're investing in the old vector if Jim Wilson has a modified one? I'm. It sounds like it must be a better vector, or no?
So that's a great point. And every time, every few months, or every, like a year, there is a better vector. But
Yeah.
when we have a better vector, like for example, if we like if we change the vector now, the work that we've done in the past year and a half, that needs to be replicated. It doesn't mean that we are not getting efficacy or safety with the current vector. The technology keeps evolving and it will always get better. Like 10 years later, it will get better than what it is today. But at the same time, the patients, including my daughter, are getting older.
Right.
So we cannot necessarily wait for, like, oh my God, this is the mind-blowing vector like that we want to use. What has been proven for safety and efficacy, what has helped in other genetic conditions, we are using that vector because the safety profile of that vector is very well tested. It is very well are captured from other genetic conditions, and we know that it works, and we know that it is the safe one. So safety first, which is why with the newer vectors which are not tested in the human patients yet, or they are still being developed, there are always these unknown questions. And basically, there's an example of this, Capsida. They had a newer capsid. Capsida as a pharmaceutical company, it showed great results in the preclinical work. But when they treated the first human patient with that new novel capsid, because of because of toxicity related to overdose, that patient unfortunately passed away. Now, the reason they're still working on identifying what the reasons are. So, one of the two reasons are potentially that, it was really uh, it worked well in the animal models which didn't have seizures. And when you go to a human patient that has seizures, the physiology of the brain is different. So, which is why potentially there was overexpression. So that is one of the working hypotheses for that company to investigate why this fatality happened in their clinical trial when they actually went from great data in animals to an actual human data. So there are many such factors, and one of the comments that came in was like, for Rett syndrome, that the first patient died because and but that's not the case. That was the first patient in UK. The first few patients in the US are actually meeting milestones, and the company had shown the 24 months data on that. So there is this important important line that needs to be drawn, that you can make this a science project and research more better vectors. You can keep doing that, but that comes at a cost of time, and these patients are just getting older. So if something has worked, it has the great safety in humans. We know that from other genetic disorders that this has worked, it is safe, it is super safe, it is effective. Then rather than making it a science experiment and keep on modifying better for better, for a better tomorrow, that better tomorrow can be like 11 years later or what have you. But what we need is treatment for these children today. So that is always what is difficult in when I work with these. For example, for the drug repurposing, when I'm working with John Hopkins and the pharmaceutical that is sponsoring this clinical trial, there is this conversation, oh, let's do a double-blinded study. I'm like, no, the patients that are enrolling with the hope that they will get seizure control. So you cannot make it a great science experiment at the expense of families that are coming with hopes of, getting treated. So if it works and if it is safe, you've seen that from other genetic therapies, and it works. So then for us for today, this is what we have, the best state of art, and we should move forward with that. Like 5 years later, definitely there will be more options. 10 years later, definitely better capsids, better, like these vectors that I started talking about. But that comes at an expense of time. And unfortunately, with every child growing older, time is not on our end. Even with, somebody getting treated today, it will be 3 months gap between the two patients. And from that perspective, once we get to first five patients, it will already be 1 years of time. So that will keep changing. That will, we need to see this in work in human patients. And based on what we've seen in other genetic conditions, we know this is the best option which is available today, the best option, which is the safest option. And then, there is this starting line that we need to cross and get to clinic, which is what's not happening in CDKL5 disorder, unfortunately. Now,
Okay.
yeah. Any
If anybody else has, I've still got questions, but if anybody else has questions, ask. I just want to ask, but then,
Yeah, I was gonna say, Kathy, you have the same questions as me. So if you
oh,
want to ask them for us.
yeah, is Lulu Foundation going to work with you with this? I mean, that guy, that oil company guy I know who funds so much of this, where is he in the funding? And
Yeah, so we've had a few discussions with the Lulu Foundation team, and we are working collaboratively to exchange data. So it's like, we are reviewing, if there is, like, for example, in CDKL5, this is a little bit of a science question, if you will. Like, we know that expression is regulated, meaning, like, even if you inject like higher dose, there is this 100% threshold and it doesn't increase beyond that.
yeah,
So, for example, these kind of questions like, okay, did you see the same information, same similar results from your animal testing? Because these two projects are very similar yet slightly different. But how it expresses in these animals, that is being like, that's how we are collaborating. We are working with each other to understand what is going on, what would be the next steps. Is this like we got this data set, does it look similar to what you've done, and so on. So that's how, like within the industry, to ensure the science questions are answered, we are collaborating that way.
because that founder can just simply write a check and, I mean, seems right. I mean,
Yeah.
that if he's on board with this, we have no financial worries.
Yes. So he did find Elaj Bio. The Lulu Foundation formed Elaj Bio, the company which is partnering it with Gemma Therapeutics, Jim Wilson's group,
Mm-hmm.
to do the slightly modified AAV9 vector to take it to the next steps. So from the Gemma and Ilaj bio perspective, this is competition. Same thing with UltraGeneX. Like for example, we work, like any scientific questions, we collaborate with UltraGeneX. But from a business perspective, it is a competition, and obviously they have much bigger funding. And over here, like a little bit background or history on me that the engineering company where I used to work for, got acquired by Walmart, and that's how I made the seed funding for this project. So yeah, going back to the question, um, from the Ilaj Bio perspective, from the uh, Ultragenyx perspective, given these are pharmaceutical companies, it is a conflict of interest. So from that perspective, that's where it gets a little more complicated than as easy as writing a check and funding this whole project.
Okay.
So there are like different pieces, like, Lulu Foundation forming Ilaj Bio and Ilaj Bio partnering up with Gemma Therapeutics. So then it becomes Gemma, like a biotech company that is working on this.
I see. Thank you. Is IFCR helping?
Yeah. So IFCR is very much helping through doctors Tim Benke, Eric Marsh, and Heather Olson. So basically, like obviously these are many different moving pieces. So it took them a little bit of time to understand what would be the best way to help all these projects. So they are forming a consortium where they are going to review the data from all these different projects, and then they're going to provide, like, these doctors are going to provide feedback. So that is where IFCR is involved.
Thank you.
Yes, please.
So you said that you guys are going to start. So if this happens by mid 2026, then the first group of kids to start is going to be 6 to 12.
Um, yes, I don't want to say any timelines because, for us as parents, like we've been burned before with these timelines. So, but going back to your question, the first group of patients or the patients that will be treated, first five are between the ages of 6 to 12 and 18 plus.
Okay. And then so like my daughter is 7 months old right now. I'm just trying to like gauge when I'm gonna know when she's gonna be able to possibly receive this if all goes according to plan. She
Yeah.
won't be able to get it until she's 2.
So most of the gene therapies first are tested or not tested. I don't want to say word tested, but are treated in children older than 2 years of age. And once we see good and safety profile across that, then only we, like as parents, as the FDA, are confident in treating it in 2 years of age or younger. So,
Okay.
so based on 7 months age and what we are expecting from the next milestones for this project, it seems highly likely that this should be ready by the second birthday.
Okay, well that sounds good.
Yeah.
Okay.
Daniel, are you working with stem cells at all?
So stem cells, we did try it on Rena, and we
Hmm.
did see good results, but those results were not sustainable. Actually, my research background is in stem cells. So when we got Rena's diagnosis, I reached out to Dr. Jean Peducci Nelson, my mentor when I was in graduate school. And the first question I was like, okay, can we use stem cells? So we did try stem cells, but those results are not sustainable beyond a few months. So from that perspective, it's a very expensive effort to keep retrying it. And unless you treat the root cause of the problem, it's going to be like, like the honeymoon period, which is commonly described.
Yeah.
These help, but for a short period of time, and then the underlying disorder kind of overtakes it. And then you don't see those benefits anymore.
But are there advances being made? I just saw a video this morning on YouTube where a little 3-year-old with Hunter's syndrome, first one cured with stem cells.
Mhm,
being
mhm.
delivered right into the brain.
Yeah.
So, are there advances being made in stem cells since you tried it, or I mean, this was an X chromosome um,
So, uh-huh.
mutation?
So,
Sorry.
so definitely there are advances made every day in different areas. I don't know of anyone specifically working on CDKL5 for stem cells. There were previously groups that were working. And obviously, it's not that everything that I know is the most updated information in CDKL5, right? Like there might be some that haven't publicly disclosed that they are working on stem cells based approach. So definitely there is a lot of like new things there. But as of today, especially what can get to clinic is not for CDKL5. There doesn't seem to be anything which is stem cell based, from at least what I know.
Thank you.
Yeah. Yeah. Uh, Any other questions? How are we doing from a fundraising perspective? What are the next steps? Anything you want me to touch on?
Yeah, do you have on there how much we're at for the fundraiser, or is it just at like gauging on like a scale? I didn't see any numbers.
Yeah, so I did put that on the Facebook group. I think the my comments also got deleted because the person who asked the questions may have deleted their questions, or at least I don't see them. So we are at 450K right now. And since that, like since all these things happened on Facebook, we haven't raised a single penny since. So it's kind of become like, a little down, but we are at 450K. And for this year, end of the year, I need to, like we need to pay Medica Bio $140,000 for them to be able to continue the project. So I was really hoping and banking on more families joining and the Thanksgiving, Giving Tuesday for fundraising, but that has since taken a huge hit. And my priority is to get to get the manufacturing material going at Medica Bio. So the 140K is what I'm trying to prioritize to raise for that because everything else was accounted for in the books and projected, and but it's not going as we were expecting for the holiday season.
Okay.
Janu, what's the end ownership thought
Mhm.
process? Like,
Yep.
typically, this type of drug is, as you said, part of a large company and Ultragenics, et cetera. What's the end goal in terms of the ownership
So that's a
structure?
great question, and thank you for that. So very similar to what other companies, or sorry, not other companies, but other foundations have done. Like for example, Fox G1, as we all know, is raising $22 million, and they are raising it for what all three phase one, phase two, and phase three, meaning all the patients with this disorder, with Fox G1 disorder, can get treatment. Now, the only thing that we are doing differently is raising for phase one and phase two. The ownership still remains with the foundation, and at phase two and the results that we get at phase two step, we then determine what the next step is for us. And to be honest, given how the funding situation has been, and with like the repetitive questions and saying that we didn't follow the protocol and those kind of comments, then so the way I am looking at it now is like we'll be as fluid, as transparent, and as open to be able to take it to the next step. Meaning, right now, this is with the foundation, the IP, the ownership, the insurance that we need to pay to UCSF when we get into the clinical trial. Everything is with the foundation. Once we get the phase one and two data, we can then open up more options, explore more options, and then see what we are doing there. Like perhaps the Elaj Bio as a a biotech company can we can have a discussion with them at that stage where we are not, like where we have human data. We can have a conversation with Ultragenics where we can support them to, like with the human based data, and they can support us and vice versa. So we, like definitely want to be open and transparent and fluid. As of today, everything is within the foundation. And, once we get the first in human data, we can explore all the options that we have on the table for us and then take the next steps from there.
And then in terms of peer review, you mentioned that was on the way.
Yes.
What is what is the process for you for that, and when do you think that will start? Because
Yep.
that's critical.
Yeah, no, definitely. It's all the data as it's been generated, it's already been peer reviewed in a sense, like, there's Terry, the SP50 father, there is Allison, there is Guy from Fox G1, there is Yael. They're all involved in this and helping out with this. So it depends on, like we're looking at a set of Mike's model data. So who are the experts in that? And then as we go on to the next steps, like, we want the clinical correlation in that. So we are, as I mentioned earlier, we are working with trying to work, through this consortium. We are working with Dr. Tim Benkei, Dr. Eric Marsh, Dr. Heather Olson, etc. So in terms of peer review, all these, for all the steps, like depending on who can provide us feedback at that step, they are involved. And then there's always the clinical translation that is going between, going in the experts of CDKL5 and going back to, like what are the next steps. So I think IFCR mentioned in January or February, they will have the details of the consortium ready. So the whole data set will also, like, along with what I am sharing right now, as and when the data comes in and sending it to them, along with that, this whole there will be this formal committee that is reviewing that, at the same as well as us working on publishing this publishing this data, and also applying for the orphan drug designation with the US FDA. So there are there are three hats, or three institutions that are looking at it from different angles, if you will, to make sure that, like, this is safe for all the patients that are going into this, and involve including myself, because I am hoping, based on the inclusion exclusion criteria, my daughter will also be one of the patients or children who can benefit from this gene therapy. Medium. Medium.
Thank you, Jane.
Thank you. I do see a few people who've just joined, so just wanted to make sure that we are going through, like, we did a discussion on what the next steps were and how we got to this step, and then we are working towards, what should we be expecting in the next steps. And this is like an open session right now where you can ask your questions, and we'll try to answer them.
Sorry if it is already answered, Jane. This is Vignesh here. Is there what are all the locations that are in US and as well as in India, like that we are targeting for?
So, as I'd mentioned in the webinar that was previously done in association with the American Society for Gene and Cell Therapy, the one location in the US right now is UCSF. And we are in talks with other institutes also, but all of these things take a lot of time. But we are trying to see if we can have multiple sites. Okay, so one of the important factors here to keep in mind are the costs associated with multiple sites, and it's just doubling up all these costs. So from that perspective, it is the focus for now is UCSF and and conversations with the other sites. I don't know if they will be ready, the other sites will be ready right at phase one, phase two. And to be honest, it seems unlikely. So those sites will be ready probably at phase three stage, both from a financial perspective and just all the paperwork and involved in the back end that includes insurances, includes the attorneys, and all of that work. Concurrent to that, we are working or planning on doing this in India at Rainbow Children's Hospital in Hyderabad. But again, that said, that the Indian regulatory landscape is little bit challenging. So that is the goal, but it may. Um, but it may vary based on as and when we see like these questions that come up. For example, from the regulatory perspective in India, the gene therapy needs to be developed in India to be able to do the trial in India. So that's sort of little bit of a new challenge that came in when we started working on the pre-IND submission there. So and there are exceptions to that rule. So we are working on that right now. But in terms of the sites, it is, the prelim sites are going to be UCSF and in the US, and in India, it's going to be Rainbow Children's Hospital. And as and when we go get closer with more data, with more financial support, with and getting through these red tape sort of hurdles, like making sure all the information for the attorneys is in place, etc. Then we are already working towards including other sites, but for now, it's not. Uh, you know, it's uh, it's not likely that they will be phase one, two sites, and it's definitely more like phase three stage.
Okay, thank you.
So, to participate in a trial, you're going to need to be very close to one of those centers, correct?
So this is one of the new pieces of information which was like recently shared, and it doesn't seem to be the case because with AADC as an example, patients travel from Poland to Canada to participate in the trial. It is ideal that you be as close as possible to that site, but that's not a requirement, and there are monitoring steps that can be done, uh, like to accommodate for the distance from the site. So so from that perspective, just having one site in San Francisco doesn't necessarily mean that somebody in Ohio or the East Coast cannot participate in it. Of course, the inclusion exclusion criteria is the determining factor on who can join and who may not be able to join the trial, and that is determined by the clinical team. That as a foundation, we don't have much of a say in that, or we don't have any say in that actually, technically.
Okay, thank you. Are you working with Cleveland Clinic?
Not at the moment.
Is there any benefit to having our child be seen at San Francisco or Colorado or Boston as opposed?
Uh, I want to say not at the moment because, definitely once it goes, the clinical trial goes live on clinicaltrial.gov, that will have the inclusion exclusion criteria and all the other details necessary. So even remotely, if a patient is a candidate like from Cleveland as an example in this question, then they can still participate in it. So the whole procedure and the protocol will be explained at that point, and there is like there doesn't seem to be any value in being proactively being seen at UCSF or Boston Children's Hospital as examples. So once this, once we get the IND approved and the protocol goes on clinicaltrials.gov, everyone will see it. And if you or us as patients can write to the site at that point, and then based on their inclusion exclusion criteria, they will ask us for more details if they need any. If not, then they will tell us whether uh, our children can be included or excluded from this trial at this stage.
Thank you.
Of course.
So for the clinical trial, the, during the clinical trial, if we have insurance through our providers, would that be like, would they be able to support that, or would they be helpful, or it's
So
not
that's a
related?
great question, and unfortunately, clinical trials for gene therapy are still at the phase where we don't have the FDA approval yet. We submit this human data, and then you get the approval. So insurance companies, at least in the US, how they work, is they only support and cover costs for approved therapeutic treatments. Clinical trials are traditionally always covered by pharmaceutical companies, and which is why, for example, when we participated with Rena in the soticlestat, it like that our cost to travel to Arizona Children's Hospital, sorry, Phoenix Children's Hospital in Arizona, that was covered, and like all the costs were covered by Takeda Pharmaceutical for us to be able to participate in the trial because you couldn't bill insurance. It's a different story that Phoenix Children's Hospital made a billing mistake and they would call us for, the bills that were past due, and we kept on saying that it is a clinical trial, and it took a little bit of time for us to have them figure it out. But that's a completely different story, and these kind of things happen in the billing department. It's very common in the US. But the existing insurance coverage will not cover for clinical trials. That's the underlying statement.
Okay, thanks. Yes, I don't know if this and if you have already answered this. Is if we have to, like, we are in Canada, if we have to participate in this trial, how long, like, are we expecting to stay in San Francisco,
Mhm,
or
mhm.
like,
So this
just
is,
for us to plan?
yep. So this is more on the clinical trial protocol itself, and the way, and this is like, obviously the FDA will approve the clinical trial protocol, so it may vary from what I am saying right now, but typically what we are foreseeing is how the clinical team is foreseeing this is 1 day of admission to the hospital. Second day will be where the AAV will be administered, or and third and fourth day are hospital stays for monitoring. And from there, monitoring at 15 days, 3 months, 6 months, at this 3 month frequency, all the way up to 3 years. Some of these steps don't I'm like can we are seeing, like, the later on, the second year and the third year steps, if they can be done remotely, but this is all for the FDA to approve, so these things may change. But as of now, if you are coming remotely, just as a, like, plan on being there for 2 weeks is what I would say.
Okay, sure, thanks.
Just a very rough like, from a parent perspective, because the child might take time to get assimilated to that specific time zone and such. So from that perspective, plan on 2 weeks, and then monitoring appointments, which can be a combination of virtual and in person at every 3 months.
Okay.
Aside from fundraising, is there any other participation required or is useful, like appealing to the FDA or appealing to companies or drug companies? Anything, any activism that we can be involved in?
So the good news is the answer is no. And I'm going to break it down into why this is good news. Because the FDA knows that CDKL5 disorder is a significant unmet need. What that means is like, they know the severity of the disorder, they know the urgency of the disorder. So it's not like, and I'm going to, give a parallel example of the Indian regulation or the FDA in India, how things are right now. So basically, in India, if we have to go do this clinical trial, we need because the work was developed here was done, all the work was done. I mean, majority of the work was done in the US. Some work was done in Spain. Some other work was done in UK. So majority of the work was done in the US. In India, this if this clinical trial needs to be done, we need to show the severity of the disorder, and that's the step that we are working on. But in the US, it's good news from that perspective that other than how we fundraise and how when we get to the IND step, like if there are any questions from the FDA and such, and the IRB, of course, IRB at UCSF, meaning the clinical team at UCSF will also have multiple questions, and how the scientific team is able to answer that, the research team is able to answer and put that package in the US, given that we've seen so many gene therapies before for other disorders, gene therapies are happening, like, with more announcements that we see each day. The FDA knows the urgency and the need to treat it at the root cause level. So, from that perspective, we don't necessarily need to show or, like to change the regulatory roadmap by coaxing the or by trying to show them the real picture of the disorder. So all of that work for rare genetic disorders has is being actively done, and the FDA sees the success in other gene therapies. So timely treatment and being able to access that treatment is important, which is in place for us. So from that perspective, we don't need to write a letter to the FDA right now explaining to them that why do we need gene therapy for CDKL5.
You?
Yes. Apart from the FDA related things, is there any other steps wherein parents, as a parent, like we could help? Like I understand for fundraise is a bigger thing, but other than the fundraise, like if we require any documentation support or like.
Yeah, that's a great question, and thanks for asking for that. So I think the one important step or the question is to have these kind of conversations, especially because there is a lot of perspective that us parents can bring to the clinical trial. So ask these questions, tell us what do you think the concerns are. My daughter is X months old. This trial may not be ready till this Y 2 years old. So this also, like, I will be taking these questions, if for some things which I don't have an answer to, I will be taking it to our scientific team and asking them, like, what do we do in these kind of questions. So definitely the involvement, the asking questions, and keep asking them till we are satisfied that we got the answer. That is very helpful in this exercise because it doesn't from no perspective it should look like this is a specific project being done in Xylos. Like, we want it to be a collective effort and we want everybody's involvement in this. So please, like, if you think your neurologist, the local neurologist, wants to see this data right now and not wait for the publication, then yes, I have the package, we put it together, we are ready to share it. We can talk to the neurologist. The neurologist can talk to directly to the scientific team, and have a back and forth discussion. So we can already get going with this, and we don't need to necessarily wait through January or February. We can already have those kind of dialogues and conversations, and more eyes that we have on this data, more eyes that we have on the clinical protocol, it will only help us plan for success for our children. So that is an important one, and like more involvement is definitely very much appreciated.
Sure. Yeah. Thank you. That was going to be my next question about like we have been seeing for our daughter here in Sikit Toronto. Um, so we were thinking about talking to our neurologist about like giving this package. Is that you have something that is readily available that
So I,
can.
I haven't put it on a Dropbox or something like that. I can work on doing that. And then, like uh, the only thing I will request is for us to know who this package is being shared with, and what is their expectation out of this package. So that is sort of important for us, because sometimes, this package has a lot of data. Like, for example, it has the certificate of analysis for the material that is coming from all these labs, right? For the materials are being manufactured. And from a clinical perspective, like that might not be relevant just to put it in front of the doctors when they only have 45 minutes to review the data. So to ensure that, like we are building it up for success, and to ensure that we are having these meaningful conversations with the neurologist and not wasting their time in the information which may be background information at that point. It will be very important to know who's reviewing this data and from what context, and then we can like make an index of the contents and point them that like this question will be answered in this document if this is what you're looking for. So it will be important to know who's looking at that data and from what perspective. So I we can start sharing it with that team accordingly. So please do let me know.
Good, thanks.
Of course. I know, like, we are around very close to time here, but I just wanted to make sure that I'm not rushing. And if there are any questions, I can still, we can extend this by another 5 minutes and take up those questions.
Yeah, I was thinking it would be nice like if we post all the recordings in the website. Yeah.
Yep. So this I will not post on the website. I will post this on the CDKL5 Facebook group, if that's okay.
Okay.
And the reason to not post it on the website is sort of like, it's very informal. And if we really think we need to post it on the website also, then I can post it. There's no reason for that. But, the website is supposed to be like more of specific to information to an audience without having the length of going through like different documents and going through different links to get one answer. So I want the website to be like, at least this is the way I'm foreseeing it, to be concise and to the point. So we can definitely post it on Facebook and YouTube channels, etc. But like if we really, as a team collectively, we really think that this should also go on the website, then totally by all means we can post it on the website.
Yeah, yeah, because like I feel like a lot of questions being asked and answered here. It's kind of a frequently asked question, right? Like we don't have to post it on the website.
Yeah, yeah, yeah,
I think like
exactly.
post it on the Facebook group, but
Of
then
course.
just update the um, website with these additional questions.
Sure.
That
Definitely. Thanks for that feedback. Definitely. That's great. Uh, so if there are no more questions, then, um, I think there's one question in chat. Let me see. Go ahead, San. It's fine. If you missed it earlier, we can definitely always answer that question again. Oh, yeah. So the question is, will the treatment only be available in the US? The answer is we are working on two sites for this One is in US at UCSF, and the other one is in India at Rainbow Children's Hospital in Hyderabad. Um, the like the path for this clinical trial in the US is very well established, and we are already working on that path. And we actually had that discussion with the FDA, where like we presented that these are the next steps that we're going to take, and they said, yeah, yes, this looks good, and this is how you should pursue it. So that pre-IND discussion was very meaningful, and that has already happened. So we have the path for the next steps. So it will be at UCSF, meaning University of California, San Francisco, as that site. And then the other one is at Rainbow Children's Hospital. However, we have not had the pre-IND discussion with the Indian FDA agency, and that's what we are working on in the next coming weeks. We have a meeting in, I think, in like 30 minutes from now or something with the Indian team, and we've had a few discussions with the regulatory CROs in India. But our what we are targeting is clinical trial at these two sites to be the first ones. Okay. Uh, yep. I think if there are no other questions, then we can stop here. And then if there is anything else, you know how to reach me on Facebook or my email or even my phone number. I think most of you have my phone number. If not, I'm going to put it in chat right now. Yep, and I look forward to any and other questions you may have and also, like if you think your neurologist would like to get involved, then like we can start sharing the data with them, and then we can take any other questions that might come up later. We can always open to taking those as well. And Giving Tuesday is coming, so please help get us to this point where we can be ready to submit the IND package. So yeah, with that, I'm going to stop recording and end today's session. And if there are any other questions, please let me know.
Thank you so much. I know.
Thank you. Okay. Thank you. I'm going to end the meeting for everybody now. So thank you. Thank you. Have a great Sunday and talk to you soon. Bye.
Bye.